Thank you very much for all the presentations from Edinburgh. The floor is open to questions, if anybody's got any questions.
I have a question. Thank you for your presentations and your very elegant mouse models. So I noticed on your KEGG pathway that you had glutathione metabolism there. I'm just wondering if you checked the glutathione levels in the liver, because it's well known that depletion can activate an adverse outcome pathway to trigger fibrosis, for example.
I'll just make sure you can hear me. Yeah, so I am really interested in that. I didn't want to focus on it because we haven't got the answers for that yet, but I was aware that we'd seen that. And there's lots of evidence of metabolic stress and oxidative stress. And what's not clear at the moment is how much of that is coming from thioacetamide and how much is coming from p53 loss, right? Mm-hmm. But yeah, and I think I pertained as well to this cluster that I didn't talk about, which was this large cholangiocyte clusters where you get this p10, p53 specific shift. So this is not tumor permissive, but this is a direct response, in my mind, to p53. And their glutathione metabolism is the main altered state. So I think p53 loss is having a big effect on glutathione metabolism. Can't speak, sorry. And yeah, I think that could be potentially a driver for ongoing oxidative stress and mutations in these cells. I think it's really, really interesting. But I'm not entirely sure how we pick that apart from the potential direct effects of thioacetamide. So I didn't want to delve into that too deep.
Thank you.
Kevin.
Hello. Thank you for the talks. They were both brilliant. This might be more of a comment than a question. I'm not sure if there's a question in here, but both P53 and P10 are sort of in genomic regions where there are multiple tumor suppressors, and the idea, and in human tumors, it's often more than just P53 that's lost. It's the whole sort of region. And I just wondered if you've thought about trying to model that in these systems and delete more than just the immediate.
I mean, we've not. So we know that just the way the transgenic is designed, that what we do is chop out a particular exon of P10 and a few exons of P53. And so we've sequenced across them just to show that we're not getting this large-scale recombination across, a big locus. But no, it's an entirely fair point. I think we're very bad at modeling these large chromosomal rearrangements that happen in cancer. And that's something we should probably consider.
Very interesting. PRH is only 3 genes along from P10. So that's why I'm asking.
Well, I've got a load of RNA-seq data on my laptop. We can have a look at what it's doing.
Yeah. And then can I ask a second quick question? Just a technical question. Is this all in male mice, or is it also in female mice?
No, we do both sexes. So we historically used female mice for the genetic screen simply because they're smaller and it works better because you require this kind of hydrostatic pressure to get the gene editing stuff into the liver. And so that was a more straightforward way of doing it. But with our transgenics, we use a mix.
Thank you, Pedro.
Hi. So congratulations for these huge amounts of data and excellent research. I was wondering, do you have any evidence if, for example, the interleukin-17 pathway is already deregulated in, for example, in precursor lesions, or, for example, in premalignant strictures in patients with PSC, or it's already happens when the tumor has already malignized in a more aggressive version?
So I think the question of is this pathway already upregulated in things like PSC is a really, really interesting one. We haven't looked at that yet. I think that's one of the next things we want to look at is, can we see any of these changes that we're seeing, interleukin-17 being one of the main ones? Can we see that in precancerous conditions such as inflammation? To try and pick apart, I guess, what effects that I'm seeing here are tumor-specific in truth, and which are part of this more inflammatory response that we're going to see in patients prior to cancer formation. So we haven't looked yet, but I definitely think one of the next steps in this research is to try and see how much this links into premalignant conditions such as PSC. In terms of one thing I've noticed as well in the data set here is that we do see some immune dysregulation in P10 and P53 loss, and not just in the tumor permissive groups, and not only when we've got tumors, but without damage. So there is some evidence there that there is immune dysregulation happening prior to damage, and therefore prior to the onset of tumor formation. But we haven't really managed to pick that apart just yet. So a short answer would be watch this space, I think.
Thanks.
Jesus.
Well, or maybe Tim, you want
Tim,
to
go ahead.
Yeah.
Okay, you got me thinking about neutrophils. So there's a
Oh, no.
no, I know. I'm sorry. There's only a couple of histological contexts that would see them in the liver. One is in the context of a large duct obstruction, where we see ductular reaction with neutrophil cholangiolytis. So in your permissive and injured, TAA injured, do they have neutrophils? And do you think in that context, PSC is one of the things that produces this histological pattern? Are you possibly modeling PSC-related cholangios? Have you got any kind of specific data on that?
Unfortunately not. I mean, it's early days, and really where we've got so far with this is an analysis of the transcriptional data. One of the first things on my mind now is to go into the tissue and look at the immune phenotype in terms of neutrophil infiltration and also kind of T-cell subsets and things like that. So I don't know whether neutrophils are there or not. If they are, I think that'd be really interesting. And I kind of suspect they might be involved, but that's just a hunch more than anything.
Okay, well, if you need some tissue, you know where to come.
I mean, I think we see some neutrophilic chemokines, right, in that data set as well.
Yeah. So we see evidence of neutrophilic chemokines, but we haven't looked specifically in situ to look for neutrophil presence.
And that was kind of what we were trying to address, I suppose, with that experiment, right? Is that what are those early sensitizing moments, be it cell autonomous or non-cell autonomous, that allows that transition from something that's inflammatory like PSC into something that's cancer? Because we really don't understand that interface at all. Particularly in cholangio, where we get tissue, that is incredibly end stage. We don't understand anything about that moment.
Following the previous question and discussion on the role of metabolism in cancer development and progression, it is well known that obesity, the metabolic syndrome, and particularly fatty liver is a risk factor for cancer, but also cholangiocarcinoma. So are you planning to stress your transgenic models to NAFLD or to, for example, drugs that induce sclerosing cholangitis like DDC and see the phenotype of independent mutation and in combination? Because I think triggering factors are missing in the model, no? Like risk factors that are well known.
Yeah, I think that's it. And I think there's, for me, I'll answer that question maybe slightly backwards by saying there's a balance between trying to model molecular mechanisms and trying to model kind of clinically relevant disease. And I always edge more towards trying to really dissect out what are the molecular pathways and players. So as you add kind of different insults and different risk factors, it gets more complicated to dissect out the biology. So I think as well as saying it would be interesting to go in that direction and model risk factors with trying to introduce kind of DDC diet or other damaging events, I'd also want to backtrack from that a little bit, pick apart more specifically the roles of P10 and P53 or individually, and what are they doing, and also dissect out damage as two separate experiments, if that makes sense. Because I feel like you can confound your analysis quite quickly by trying to be too clinically representative in one fell swoop, if that makes sense.
There is one paper just in Press in Hepatology showing that the most proliferative and aggressive cholangiocarcinoma cells demand more free fatty acids and lipoproteins for the growth and for the promotion of the dissemination. So I think that would be very interesting.
I think certainly putting it in a fatty liver would be a really good idea. I differ from Ed in this. I'm just like, more models.
I think we counterbalance.
Yeah, we're very well balanced.
Thank you. Yeah, Izzy.
Thank you. That was really great. I have a question for Ed. Actually, first, sorry, I don't know your name. I'm really sorry.
Pedro.
In terms of Pedro's question about pre-malignant lesions, there is a paper that has shown that in biliary atresia, you do get increased IL-17. Okay. Which is, I can send you the link, which is really interesting because I think that the IL-17 signal comes up quite early in terms of biliary injury and might have something to do with regeneration as well. But my question was about, as I said before, I'm a fan of spatial analysis, basically. And it's been shown that neutrophils, apart from their role in wound healing, have quite a big role in tumor initiation and progression in certain cancers. And secondly, some studies have shown that neutrophils form a relevant source of IL-17 in certain models. So I just wondered whether you'd considered kind of doing multiplex staining, where you can stain for different cell subsets within your mouse tissue. So you can stain for neutrophils and macrophages and gamma delta T cells, because those are quite highly represented in the mice, more so than in the human, which is why the mouse is such a great model for looking at those cells. And you can potentially, with, I'm not going to say simple, because nothing's ever simple, but you can potentially pull apart the source of your IL-17 and what it's doing within your model.
Yeah, I think that's really important. That's definitely next steps for this. I think for me, I was working on the lines of probably there being gamma delta T cells or Th17 T cells there. That would be the main source of interleukin 17, but we don't know this. So I think teasing apart where the interleukin 17 is coming from will be a really, really interesting step. The fact that you say you see this upregulated in biliary atresia is interesting, because we don't see it, the interleukin 17 signature, in the damage alone group. So then you've got this question of, are we using the wrong damage stimulus? If in practice what we see is interleukin 17 upregulated in the patients as a preemption of cancer happening prior to cancer, then we're not quite modelling that here. So we personally need to be careful of that. I think, we're going to try and understand where interleukin 17 is coming from in our model, what immune cells are involved. But I think a next step after that is then to really see how interleukin 17 looks in patient samples or pre-malignant diseases, and just not presume that we're representing that perfectly here. I think that's important.
Thank you.
Thank you.
The frequency of mutations in KRAS and P53 are higher in perihilar and distal tumors compared to intrahepatic. Your experimental models also reproduce development of tumors along the biliary tree, or are mainly intrahepatic due to the...
Yeah, so all the hydrodynamic ones are intrahepatic, obviously, because that's where you deliver the kind of mutations. Yeah. So for the transgenic, the way the keratin 19 CRE works is that it preferentially recombines in small ducts. So that's where we delete P10 and P53. There is, and I was talking to Simon about this over coffee. There's a mouse that came out in Cancer Discovery earlier this year, where they express a catalytic mutant of PI3 kinase in the extrahepatic duct, and that gives you at least the early phases of extrahepatic cholangio. And so I think that's something that we as a community lack, right? A really good transgenic model of hilar or distal.
Just on a related note, as you know, you mentioned the fact that we're doing the clinical trial with a Wnt inhibitor. I know your focus has been on intrahepatic cholangiocarcinoma, but do you think Wnt and PI3 kinase are also hyperactivated in perihilar and distal? Do you have any sense of that?
Not formally, although kind of based on the literature, there would certainly for PI3 kinase, I think you're seeing extra the kind of distal mutations in PI3 kinase in the Liverpool data set. There's this mouse, so I think certainly there. Wnt-wise, it wouldn't surprise me that there was Wnt activation there too.
Thank you. Thank you. If there are no more questions, I'd like to thank our speakers from the Scottish team.